There is a plant growing on a rocky hillside somewhere on the edge of the Tibetan Plateau, roughly 12,000 years ago. It is about a meter tall, weedy, thin-stalked, covered in tiny hairs, and it smells. It smells strong enough that you can catch it downwind from 30 paces. The seeds are small, dark, and fall off the stem the moment they ripen, scattering themselves across the ground to reproduce without anyone's help. Nobody planted it. Nobody bred it. It is growing exactly the way it has been growing for millions of years, minding its own business in the foothills of Central Asia. It is one of the most unremarkable organisms on the planet. Fast forward 12,000 years, and that same species has been split into two completely different plants that barely resemble each other. One grows 5 meters tall, thin as a broomstick, produces almost no resin, and is harvested for rope and fabric. The other is short, bushy, dripping with sticky crystals, and contains a chemical so potent it alters the way your brain processes reality. Same species, same DNA, shaped by humans into two things that could not be more different, that did not happen by accident. That is one of the most dramatic feats of selective breeding in the history of agriculture. And almost nobody knows how it actually happened. If you enjoy this kind of content, drop a like on this video. It genuinely helps. Now let's get into it. So, how did we take a wild weed and turn it into one of the most versatile, most controversial, and most heavily modified plants on Earth? The answer starts not with humans, but with the plant itself. Cannabis belongs to a botanical family called Cannabaceae, and its closest living relative might surprise you: hops. The plant that flavors beer. Cannabis and Humulus lupulus share a common ancestor. In 2019, botanist John McPartland published a study in Vegetation History and Archaeobotany that used fossil pollen records to estimate when the two lineages split. His answer, roughly 19.6 million years ago on the northeastern Tibetan Plateau. That means cannabis as a genus has been around since before humans existed, since before the genus Homo existed, since before the great apes existed. This plant was here first by a wide margin. Humans are the newcomers in this relationship, and here's where this connects to domestication. When we eventually found this plant, it was not blank raw material waiting for us to do something clever with it. It had already been shaped by millions of years of natural selection into something very specific. A fast-growing, wind-pollinated annual that thrived in disturbed soils, open ground, the kind of nitrogen-rich waste areas that form around animal herds and, eventually, around human camps. It was already a weed in the most literal sense. It grew where we dumped things, and that is probably how we met it. Think about that for a second. We did not go searching for cannabis. Cannabis came to us. It showed up in our garbage heaps, our latrines, the clearings we made when we cut down trees and burned brush. Nitrogen-rich, sun-drenched, disturbed soil. That is cannabis paradise. And by the time humans started settling down in East Asia around 12,000 years ago, this plant was already thriving in the biological shadow of every campsite and village. Now, here's the question that matters. When and where did we actually domesticate it? For decades, the assumption was Central Asia. Nikolai Vavilov, the legendary Soviet botanist who spent the 1920s and 1930s mapping the geographic origins of the world's major crops, placed cannabis in a broad Central Asian zone stretching from the Caspian Sea to the mountains of western China. That became the textbook answer for almost a century. Then in 2021, a team of researchers led by Liaoming Ren at the University of Lausanne and the Kunming Institute of Botany published a study in Science Advances that rewrote the story. The team sequenced 110 complete cannabis genomes, the largest genomic study of the plant ever conducted. They sampled everything: wild plants, feral populations, ancient landraces, modern hemp cultivars, modern drug cultivars, specimens from 6 continents. And when they mapped the genetic relationships, the family tree pointed to a single place: eastern China, the northeastern edge of the Tibetan Plateau, Qinghai and Gansu provinces. That is where the oldest, most genetically diverse cannabis populations still grow today. Those feral plants in the Chinese foothills are the closest living relatives of the original wild ancestor. Everything else, every hemp field in France, every coffee shop strain in Amsterdam, every rope ever pulled on a sailing ship, descends from that population. One starting point. One domestication event. Roughly 12,000 years ago, right at the beginning of the Neolithic. Think about what that timing means. Cannabis was domesticated at the same time as the very first crops on Earth. Einkorn wheat in the Fertile Crescent around 10,500 years ago. Barley around 10,000. Rice in the Yangtze Valley around 9,000. Foxtail millet in the Yellow River Basin around the same time. Cannabis is not some latecomer that humans picked up after they had already mastered farming. It was there from the start, one of the founding crops of the entire agricultural revolution, at least in East Asia. The question is, why this plant? Because it could do everything. This is the part most people do not fully appreciate. Wild cannabis was not useful for just one thing. It was useful for at least 4 completely separate things, and early humans figured out all of them. First, fiber. The stalks contain long, bast fibers, tough, flexible strands that can be twisted into cord, woven into fabric, or braided into rope. Hemp cord impressions have been found on Neolithic pottery in China and Japan dating back roughly 10,000 to 12,000 years, some of the oldest evidence of textile technology anywhere on Earth. Before cotton, before wool was widely used, before linen from flax spread across the Near East, people in East Asia were making rope and cloth from cannabis. Second, food. Cannabis seeds are roughly 30% oil and 25% protein by weight. They contain all essential amino acids. Hemp seed oil could be used for cooking, for lighting lamps, for waterproofing. In a world where calories were scarce and fat was survival, a plant that produced oil-rich seeds by the handful was not a luxury. It was insurance. Third, medicine. The oldest surviving reference to cannabis as a medicinal plant comes from a Chinese text called the Pensao Qing, traditionally attributed to the legendary Emperor Shennong. The text itself was probably compiled around the 1st or 2nd century AD from much older oral traditions, but it recommended cannabis for joint pain, constipation, malaria, and what it described as absent-mindedness. Whether or not Shennong was a real person, the tradition of using cannabis medicinally in China stretches back thousands of years. And fourth, the one everybody already knows about. The plant produces a resin. That resin contains a molecule called delta-9 tetrahydrocannabinol, THC. And THC does something to your brain that almost no other naturally occurring chemical does. It plugs into a receptor system that your body already has, a system built around a molecule you produce naturally called anandamide, named from the Sanskrit word for bliss. Your brain has a lock. Cannabis has a spare key. And somebody a very long time ago figured that out. But here's the thing that connects all 4 uses to the domestication story. You cannot optimize a single plant for all 4 at once. A tall, straight-stalked plant that produces long fibers is a terrible drug plant. A short, bushy, resin-drenched plant is useless for making rope. A plant bred for seed production is different from a plant bred for fiber length or THC content. The moment humans started selecting for specific traits, they started pulling this one species apart into multiple directions. And that is exactly what the genetics show. Wren's 2021 study found that the split between hemp-type and drug-type cannabis happened roughly 4,000 years ago, around 2000 BC. Before that, for about 8,000 years, humans appear to have been growing cannabis as a general-purpose crop, selecting casually for seed size, for faster growth, for the basic domestication traits that make any wild plant easier to harvest, seeds that do not shatter and fall to the ground the moment they ripen, stems that grow taller and straighter, faster germination, less dormancy, the usual domestication syndrome that shows up in wheat, barley, rice, and almost every other crop humans have ever tamed. But around 4,000 years ago, the selection pressure split. And the genetic evidence for this is specific enough to be genuinely interesting. Cannabis produces its cannabinoids through a biosynthetic pathway that starts with a precursor molecule called CBGA, cannabigerolic acid. From that single precursor, 2 competing enzymes go to work. One is encoded by a gene called THCAS, THC acid synthase, which converts the precursor into THCA, which becomes THC when heated. The other is encoded by CBDAS, CBD acid synthase, which converts the same precursor into CBDA, which becomes CBD. Here is the critical detail. Those 2 genes sit at the same location on the same chromosome. They are allelic variants competing for the same slot. It is a molecular either-or. A plant that expresses a lot of THCAs produces a lot of THC and very little CBD. A plant that expresses a lot of CBDAs produces a lot of CBD and very little THC. This is the genetic switch that separates hemp from drug cannabis. And humans, without knowing any of this biochemistry, pushed that switch in both directions simultaneously. In drug cultivars, Wren's team found that the THCase gene had been amplified, multiple copies, increased expression. The plants were flooding the biosynthetic pathway with the THC enzyme. In hemp cultivars, the opposite had happened. THCase was reduced or non-functional. CBDAs had taken over, same species, same genome, same chromosome. Two opposite outcomes, both driven by thousands of years of people picking the plants they liked best and replanting the seeds. Wild cannabis produces THC at roughly 1 to 3% by dry weight. Modern drug varieties can hit 25 to 30%. That is a tenfold increase achieved entirely through selective breeding. Modern hemp, by legal definition in most countries, contains less than 0.3% THC. That threshold, by the way, was set in 1976 by a Canadian botanist named Ernest Small, who published a paper proposing the 0.3% line as a practical cutoff to distinguish hemp from drug cannabis. It was a somewhat arbitrary number based on the distribution of THC content he observed in different populations. Governments adopted it as law. A botanical observation became a legal boundary. But the domestication story is not just about chemistry, it is about architecture. And this is where you can literally see what humans did to this plant. Wild cannabis is a moderate-sized annual, maybe 1 to 2 meters tall, with a natural branching pattern that distributes its energy across multiple stems and seed heads. Humans pulled that shape in two opposite directions. For fiber, they selected for plants that grew tall and straight with minimal branching. A single thick stem as long as possible with long internodes because the fiber comes from the bark of the stalk and longer stalks mean longer fibers. Modern hemp can grow to 5 meters, over 16 feet in a single growing season. That is not natural. That is thousands of years of farmers looking at a field, picking the tallest, straightest plants, saving their seeds, and replanting the next year. For drug production, they selected for the opposite. Short, bushy, heavy branching, maximum flower sites. Dense clusters of resinous buds on every branch, and eventually the technique of sinsemilla, growing only female plants and removing all males so the females never get pollinated. Unpollinated female cannabis plants produce dramatically more resin than pollinated ones because the plant keeps producing trichomes, the tiny resin glands, in a prolonged effort to catch pollen that never arrives. That technique was refined significantly in the 20th century, but the basic principle that unfertilized females produce more resin is something growers may have understood for centuries. Now, here's a pattern interrupt. Stop and think about what this means in the larger context of human agriculture. We talk about the domestication of wheat and corn and rice as the great achievements of early farming. And they were. But cannabis might be the only plant in history that was simultaneously domesticated for fiber, for food, for medicine, and for its effect on consciousness. No other crop does that. Cotton gives you fiber but not food. Wheat gives you food but not fiber. Poppies give you medicine and altered consciousness but no rope. Cannabis did all of it. It was the original multi-purpose crop, the Swiss Army knife of the plant kingdom. And the fact that we eventually split it into specialist varieties tells you something about how human agriculture works. Generalists get replaced by specialists. Every time. So when did this split plant start traveling? The pollen and archaeological record tells that story in surprising detail. Teng Wenlong and colleagues published a study in 2017 in Vegetation History and Archaeobotany compiling palynological data from 143 sites across Eurasia. Cannabis pollen shows up across China starting roughly 10,000 years ago, with a significant increase around 5,000 years ago as agricultural intensification picked up. By around 5,000 to 4,000 years ago, it was spreading westward along what would become the Silk Road across the Central Asian steppe, carried by the same nomadic herders and traders who moved horses, wheat, and bronze technology between east and west. And here is where the domestication story meets the archaeological record in a genuinely dramatic way. In 2008, neurologist Ethan Russo and his team published a study in the Journal of Experimental Botany describing the contents of Tomb M90 at the Yanghai burial site in the Turpan Basin of western China. Inside a 2,700-year-old grave belonging to a Gushi culture shaman, they found roughly 789 grams of cannabis, almost 2 pounds, and this was not raw, unprocessed material. The seeds and stems had been carefully removed, leaving only the flowering tops. Chemical analysis confirmed the presence of THC. This was cannabis cultivated specifically for its psychoactive properties, processed with obvious expertise, and buried with a man whose community considered it valuable enough to send into the afterlife. 2,700 years ago, someone was already growing high-quality drug cannabis, harvesting it with care, and treating it as something sacred. 500 years later, roughly 500 BC in the Pamir Mountains along the border of modern China and Tajikistan, people at a site called Jirzankal Cemetery were burning cannabis on heated stones inside wooden braziers during funeral rituals. In 2019, Yimin Yang of the University of Chinese Academy of Sciences led a team that analyzed the residue from those braziers using gas chromatography mass spectrometry. The cannabis had significantly higher THC levels than wild plants growing in the same region. These people were not burning whatever happened to grow nearby, they were selecting, possibly cultivating high-potency cannabis for ritual use, and they were doing it at high altitude over 3,000 meters where UV radiation is more intense, and cannabis plants naturally produce more THC as a protective response. The mountains themselves were part of the domestication process. Geography and human selection working together. And by this same period, the Greek historian Herodotus, writing around 440 BC, was watching Scythian nomads on the Central Asian steppe crawl inside small felt tents, throw cannabis onto red-hot stones, and breathe in the smoke as a funerary ritual. He described it in the Histories, Book 4, "They howled with delight." For a long time, scholars assumed Herodotus was exaggerating. Then Soviet archaeologist Sergei Rudenko excavated the frozen Pazyryk tombs in the Altai Mountains of Siberia, starting in 1947, and found exactly what Herodotus described: tent frames, heated stones. Cannabis seeds, preserved in permafrost for over 2,000 years. The Greek historian was reporting what he saw. By this point, roughly 2,500 years ago, domesticated cannabis had spread across the entire breadth of Eurasia. From China to the Caucasus, from India, where the Atharva Veda, compiled between roughly 1500 and 1000 BC, lists cannabis as one of 5 sacred plants, to the eastern Mediterranean, where the Assyrians called it kunabu, the word from which cannabis itself derives, and recorded its use as a fumigant and medicine in texts from the library of Ashurbanipal around 660 BC. And in 2020, a team led by Aaron Ari published a finding in the Tel Aviv Journal of the Institute of Archaeology that added another layer. At Tel Arad in the Negev Desert, a shrine dating to the ancient kingdom of Judah around 750 to 715 BC had 2 altars. One was burning frankincense. The other was burning cannabis mixed with animal dung. The dung used to control the burn temperature and release the smoke slowly. Somebody in an ancient Judahite temple understood combustion chemistry well enough to optimize the delivery of THC from a plant to a room full of worshippers. That is not casual use. That is technology applied to ritual 2,700 years ago. Now, while all of this drug use history was unfolding, the fiber side of the domestication story was just as consequential, if less glamorous. Hemp rope and fabric have been found at archaeological sites across China, Japan, and eventually Europe, spanning thousands of years. By the medieval period, hemp was one of the most important industrial crops on the continent. Rope, sailcloth, canvas, a word that comes from the Latin cannabis. The entire age of exploration, every ship that crossed the Atlantic or rounded the Cape was held together by hemp rigging. When European colonists arrived in the Americas, they brought hemp seeds with them. The Virginia Assembly passed a law making hemp cultivation mandatory for farmers in 1619. Not optional, required. That is how important fiber hemp was to the functioning of early colonial economies. And here is where the 2 domestication stories, fiber and drug, start pulling the species even further apart. The fiber growers wanted tall, straight, uniform plants with as little resin as possible, because resin makes processing the fiber messy. The drug growers wanted the opposite: short, branchy, resin-soaked, and because cannabis is an outcrossing species, meaning it relies on wind to carry pollen between separate male and female plants, isolation was critical. If your drug field is downwind from a hemp field, the pollen crosses and your offspring are somewhere in between. Not ideal for either purpose. Over centuries, this pressure drove the 2 types further apart geographically and genetically. Hemp cultivation centered in Northern Europe and East Asia. Drug cultivation centered in South Asia, the Middle East, and parts of Africa. Geographer Barney Whorf at the University of Kansas traced these migration routes in a 2014 paper in the Geographical Review titled High Points, A Historical Geography of Cannabis. His mapping shows cannabis reaching sub-Saharan Africa via East African trade routes and Arab merchants roughly 1,000 to 500 years ago. It reached the Americas with European colonists in the 16th century. By the 20th century, it was growing on every inhabited continent. One wild plant from the foothills of the Tibetan Plateau had, with human help, colonized the entire planet. And then something happened that had never happened in 12,000 years of coevolution. We tried to uninvent it. In 1930, a man named Harry Anslinger became head of the U.S. Federal Bureau of Narcotics and launched a campaign that systematically tied cannabis to racial minorities, fabricated links between the plant and violent crime, and rebranded it with the Spanish word marijuana, specifically to make it sound foreign and threatening. In 1937, the Marijuana Tax Act effectively criminalized the plant. In 1970, Richard Nixon's Controlled Substances Act placed it on Schedule I, the same classification as heroin, defined as having no accepted medical use and a high potential for abuse. And the consequences were global. The 1961 UN Single Convention on Narcotic Drugs, pushed by countries including the United States, placed cannabis under international restrictions that affected every signatory nation. A plant that humans had been cultivating for 12,000 years, that had been woven into the sails of ships, ground into medicine, burned in temples, and bred into dozens of specialized varieties across 6 continents, was declared too dangerous to exist in approximately 0.7% of its relationship with our species. But prohibition did not stop the domestication. It just moved it underground, and arguably, it accelerated it. In the decades after criminalization, illicit growers, working in basements and closets under artificial lights, applied selection pressure to cannabis at a pace that would have stunned any Neolithic farmer. They crossed Afghan indica varieties with Southeast Asian and South American sativas. They selected obsessively for THC content, flavor, growth speed, and compact size. They developed hydroponic and indoor growing systems that gave them total control over light, nutrients, and flowering schedules. In 2 generations of underground breeding, they pushed THC levels from the 3 to 5% range common in the 1970s to the 20 to 30% range common today. That is a 6 to 10-fold increase in potency in roughly 50 years. It took thousands of years of traditional agriculture to get cannabis from wild-type THC levels to 5%. It took modern Prohibition-era breeders about half a century to triple or quadruple that number. Illegality, it turns out, is an extraordinarily effective selection pressure. Now, here's the part that answers the question you came here for. How did humans domesticate cannabis? The same way we domesticated everything else. Slowly, then all at once. We noticed a useful plant growing in our waste piles roughly 12,000 years ago in the foothills of eastern China. We started saving seeds from the plants we liked best. Taller ones for fiber, oilier seeds for food, more resinous flowers for medicine and ritual. Over thousands of years, those preferences compounded. The genes for THC production and the genes for CBD production sat on the same chromosome, competing for the same molecular machinery, and we pushed them in opposite directions without understanding a single thing about the underlying biochemistry. We bred hemp that grows 5 meters tall and contains almost no THC. We bred drug cannabis that barely reaches your waist and contains 30 times the THC of its wild ancestor. We moved it across every continent, adapted it to every climate from the tropics to Scandinavia, integrated it into religions, pharmacopeias, navies, economies, and criminal justice systems, and then we tried to ban it, which only made the breeders work faster. Is cannabis domestication a single story from a single origin? According to the best available genetic evidence, yes. The 2021 REN study's whole genome analysis points to one domestication event from one ancestral population in eastern China that then branched into hemp and drug lineages around 4,000 years ago. The probability of a single origin is high, roughly 85 to 90% based on the genomic convergence of all modern varieties toward that one basal population. The probability that wild-type cannabis still exists anywhere on Earth, untouched by human selection or hybridization, is close to 0. REN's team found that virtually every wild population they sampled showed genetic signatures of past contact with domesticated varieties. We did not just tame this plant, we replaced its wild ancestors so completely that the original unmodified version may no longer exist. And that, honestly, might be the most telling detail of all. We found a weed growing in our garbage 12,000 years ago. We liked what it did. We kept the seeds. We replanted them. We picked the ones that did it better. And we did that for so long across so many generations on so many continents that the original plant, the version that existed before we ever touched it, is gone. Not extinct exactly, just absorbed, overwritten, replaced by the versions we preferred. Tomorrow, somewhere on this planet, a farmer will plant a hemp seed that will grow 16 feet tall and produce fiber strong enough to reinforce concrete. And somewhere else, in a basement or a greenhouse or a licensed facility, someone will tend a squat, crystalline plant bred to produce 30% THC by dry weight. Both of those plants are the same species. Both carry the same genome. And both are the product of 12,000 years of human decisions, most of them made by people who had no idea they were running one of the longest genetic experiments in history. They just knew the plant did something worth keeping, and so they kept it. Now, if you found this interesting and learned something new, smash that like button and share this video with someone who would enjoy it. Subscribe if you haven't already, and drop a comment telling me what topic I should cover next. And if you want more, the next video is already waiting for you right there on your screen.